Okay, let's, I guess, maybe start to get going to breed good behavior for starting on time. First of all, are there any questions from last lecture about any logistical matters, like how the course is organized, how it's being graded? Any questions about course logistics, one way or another? Okay, that's good. Okay, any questions about any technical material from last time? You know, anything about DNA or about biology, why it's good, anything that was confusing, anything that people, if they thought about it, came up as starting to be interesting. Yes? Wait, say that again? Right. Okay, so the question is, why is it, as I understand the question is, why is it that there are four, that the coding system of nature is triples of DNA? Remember, what was the basic issue? The basic issue was that you need the code for 20 different amino acids, okay? And, in principle, actually, there's another symbol called the stop codon. So from an information theory point of view, all you need, really, is enough bits to code for 21 possibilities, which would be how many bits? Okay? That would normally be 5 bits, right? 5 bits, because 2 to the 32 is equal, 2 to the 5 is equal to 32, right? So in principle, if we wanted to redesign life all over from scratch, we could say, forget about this base 4 alphabet of DNA, okay? Let's start with a binary alphabet. It worked well with computers. Maybe it would work well with life, okay? The truth is that we are not starting life from scratch, okay? Life is something we are given. And nature evolved, basically, the genetic code the way that it did, okay, for reasons that are somewhat mysterious today, okay? So what I would say is the fact that DNA is a 4-letter alphabet is a function of chemistry. It's how nucleic acids work, okay? And once you have a 4-letter alphabet, it becomes clear that you've got 4 times 4, 2 bases would not be enough. That would only code for 16 possibilities. So that is an argument as to why, nature, if it wanted to use 20 amino acids in proteins, had to evolve a 3 base code. Okay? Does that answer your question? Okay. So some of this is historical, I won't say accident, but somehow life evolved and it's here. And the issues of how the coding translation works is very, very fundamental stuff. I mean, essentially all organisms on Earth use the same coding table, which is kind of an amazing thing if you think about it. It means that you have something very fundamental that you share, which seems very complicated, this coding table. You share it with every bacteria, you share it with every mammal, every bird, every fish. So it's something that is fundamental chemistry that somehow evolution has had a hard time playing with, okay? So it's kind of gotten locked in and is an important thing. And it's probably because the chemistry of DNA is a complicated thing. In general, here we're going to ignore details of chemistry as much as possible. But the thing that is interesting is to recognize that a lot of the reasons why life is what it is because of how molecules work, okay? And it's not for me to redesign chemistry to make it better, okay? Any questions on this or any related subject? Okay? Any questions about DNA, what we talked about last time, or course logistics, or anything like that? Okay. The question, actually, a couple of things about the question. First, I heard, how many people listened to the tape? Okay, a few people listened to the tape to prove that it worked. Um, I was told that I talked too loud, so I attempted to move my microphone down. Yeah?
I might suggest actually put it on your belt because I can still hear my voice when I spoke. So if you move it down further, then it would be more in line with the two students talking.
Okay, what we're going to do now is I'm going to keep it down here. If I'm too loud again, come back and tell me I'm too loud, and I'll lower it even more. And you'll see how far I can put this down over the course of the semester. One other question that did come out, though, is that student questions don't appear on here. And so if there's anybody who asks a question, the rest of the class, if I don't repeat the question, some of you may say repeat the question, and that would be good for future generations. Okay? Any questions? Okay? So any questions about DNA or the course requirements or anything like that before I get into the lecture for today? Okay, so the question that we had was about the evolution, and sometimes it was a question of why is the genetic code, this triplet code, the way that it is, okay? And the real answer to that, and the real answer to almost anything, if you ask why is something like this in biology, the usual answer as to why is the answer is it is evolution, okay? And what is evolution? I mean, all of you, I suspect, have heard of Darwin and the theory of evolution. It's a very, very powerful thing that explains a lot of forces in how life is what it is. And it works basically, there's basically two parts of it. The whole idea of evolutionary theory is based on the fact that somehow there are changes. Each organism's genomic sequence somehow is different than that of its parents in some way. Okay? One way might be because of mutations. Okay? A common thing, what is a mutation? Actually, this is bad. What I just did was very, very bad. Okay? Very, very bad. Sorry about that. Okay? This is one that this one should get on YouTube, I guess. If I'm not careful about that. Okay, sorry about that. This goes away, and don't let me do that again. Okay, so what is there about evolution? Okay, so we have talked about how there is a genomic DNA sequence. You are defined in some sense by your genomic DNA sequence. In the course of making this DNA sequence, a couple of different things happened. Okay, one is in some sense you are all a mix of your parents. Okay, that much I think everybody understands. And so somehow there is some mechanism that I don't want to go into too much in detail, but I'll call recombination, okay, that basically says that you are a mix of your parents' DNA sequence, okay? Exactly how you're a mix of it, I don't want to go into too much detail, but basically I guess it would say that your genome sequence can be thought of as being parts of it come from mommy, parts of it come from daddy, and it sort of mixes it back and forth. And so you're made up of large hunks of mommy followed by large hunks of daddy, okay? And we'll go into this, I think, a little more detail later. So one source of variation is this thing called recombination, and that's an important phenomenon. Another source of variation is mutations, that every time a cell divides, okay, the genomic DNA has to be copied. If you think about it, that's 3 billion bases of information. Every once in a while, you would expect there to be a copying error. Okay, if I had to do a chemical reaction 3 billion times, the idea that it's going to happen perfectly each time is just isn't true. In fact, it's amazing how accurately it does happen, and the cell has very complicated machinery to try to copy DNA effectively and correct errors and things like that. But what we call a mutation is a rare event where sometimes there is a single base change. So for the most part, you are a copy of a mix of mommy and daddy with a couple of errors introduced. Okay? That is how I'm going to say sort of your genetic material is for now. Okay? And as far as evolution goes, you're now a different person. You're different than every other person in the room. Okay? If you are a mix that is better at surviving, okay, or better at having children, you're more attractive, you have more babies somehow, odds are your changes will get passed on to future generations. And as time goes by, the favorable changes should accumulate, okay? You could imagine that when you make these copying mistakes, okay, usually it will, well, if you think about what happens when there is a DNA copying mistake. If we believe that the DNA was 2% genes and 8% junk, what would happen most of the time when there is a mutation? Okay? Nothing, right? It copies, boilerplate. It changes a C to a T and nothing happens, okay? So there's a lot of what we call synonymous or silent mutations, and it's no big deal. Every once in a while, there will be a mutation that will affect something that is very important, and usually it'll be disadvantageous. Maybe you'll change a base in a protein, in a gene, and that may cause the amino acid to change, and there's going to be a slightly different chemical composition of this protein. And usually this is a bad thing, and maybe now you'll be sick, you won't have children, you won't live to a long age, mutation will die with you, okay? But occasionally, something good will happen, and it will make a more useful protein, and you will, will make you suddenly want to have lots of children, okay? It may make you live longer, okay? Somehow, if it's a good thing, it is more likely to be selected for, okay, and accumulate. And over time, somehow, through this process of natural selection, good changes tend to accumulate. Neutral changes probably accumulate, but who cares, okay? And, through a drift process, that's what they would say the neutral changes are. And bad changes, there was a try, but didn't work, and that doesn't get passed on. Any questions about that? Yes? So the genome, the question of how much do the question is really now about how much do the genomes of different people differ. The answer should be clear that everybody's genome differs. If we look around this room, all of you look different, okay? So there is something different between me and you, okay? And that something is somehow reflected in our genetic sequence. Now, the question of how different we are is maybe the more interesting question. And the answer is not very much, okay? So if we think about it, I have a slide about this later, but I mean, basically, roughly, if we take what is the difference between my 3 billion bases and your 3 billion bases, we differ in approximately 1 out of every 1,000 bases, okay? So the answer of how similar are we, okay? The answer is quite similar, okay? That's why most things that happen to me basically will happen to you, okay? Well, that's why we had food tasters. Remember food taster? In the old days when you were a king, you were afraid of being poisoned, okay? You would have a subject, you say, you eat my food first, okay? Why did that work? It was because if this other guy was likely to be poisoned by the food as well as you were, right? Because they were similar enough that they would react biologically the same, okay? So the basic point is that within a species, there is variation. That variation is seemingly small, but it's big enough to capture all the changes in this room and the difference of how we look, what diseases we might have, things like that, okay? So the answer is we're quite similar, but we're not completely similar. And for some things, we want to be thought of as similar enough that it doesn't matter. And for in some other contexts, the differences matter. Okay? Any questions? Yeah.
So when we say human genome sequence,
Right.
so that would correspond to a specific person's genome?
Okay, so this now gets to the question of when we say the human genome was sequenced, whose genome was it? That's really what you're asking. And on one level, the question now is, does that matter or does that not matter? Okay? If you believe me that you differ from that person in only 1 out of 1,000 bases, how much does it matter? Well, whoever it was, that person is what, 99.99 or 99.9% the same as you, right? So you could think of it as a human genome was sequenced, okay? But that provided a tremendous amount of information about you. When they sequenced the first guy, okay, let's call him Craig, okay? Craig was 99.9% the same as you. So they sequenced 99.9% of you, okay, and that's very good. Now then, as you study more and more, you start worrying about things like what about the human variation? And so a lot of what is going on right now in genomics is about understanding the differences between people. But before we get into that, we should step back and recognize all the similarity. Okay, that's sort of most important. Any questions? Any of this? Okay? So bottom line, evolution is an important thing for us. Let's go and boom. Let's go. I think it's this one. No, I think I lost it already. Okay. Boom. So one reason why evolution is important to us is going to be very important. We're going to talk a lot in here about string searching, okay, or string comparison. We have this vision that these ACGT genome sequences are important, okay? One reason they're important is that we're going to be doing operations on them, like comparing them, okay, and seeing what's similar, what's different. Homology is the fancy word we'll use here for similarity among sequences. So two DNA sequences are similar. If they're similar, they have a high homology, okay? And what's interesting is that this idea of evolution, okay, motivates the idea that I should learn something by comparing similar sequences, okay? So let's say we take a look at the beginning, okay? If we look at species, we have sort of this evolutionary theory. Somehow at the beginning there was a first life form. And through various processes of recombination and mutations, various other life forms split off, and you wait 100 million years, or 100, however many years since the first event, okay, and now you've got a bunch of species living today. Humans and chimpanzees split off about 5 million years ago. Okay, that's when there was the last common ancestor between humans and chimpanzees. Okay? Now, humans and mice split off about 100 million years ago. Okay? Is my understanding. Okay, that's where there was a last common ancestor here. So now, what would we expect, let's say, between how our genes are similar to genes in chimps? Okay? There has been only 5 million years of mutations and recombinations happening. Okay? We would expect that if that's a short enough time, the gene for, let's say, hemoglobin, an important protein, in blood, you've probably heard of hemoglobin. Okay? The hemoglobin gene in monkeys should be quite similar to that of humans. Okay? Probably not identical. We're more different than monkeys. Okay? We're probably differ between us humans and monkeys in 1 out of every 100 bases instead of 1 out of every 1,000 bases. But still, you could imagine that because there is this evolutionary origin, we're both descended from the same, 5 million years ago we had the same ancestor. Since then, we and chimps have both been accumulating mutations. But you would still expect that if you want to find what the hemoglobin gene is in humans, it's going to look quite a bit like the one in chimps, okay? And this is why we think that doing sequence comparisons will provide us useful information. If we are descendants of a common ancestor, and the sequences that we see in two genomes are similar enough, they probably have the same function, they have the same origin, and that can tell us a lot about what that gene does, okay? So let's say that we look at mice. We find that there is a gene that seems to affect mice getting fat. Okay, they construct mutations of mice. They find that there's a mouse that really likes to get fat. Okay, they find what gene is responsible for it in mice. We would expect that by taking that DNA sequence and doing a search in the human genome, there probably is a similar gene because only 100 million years ago we had a common ancestor. And it now starts to become interesting is what is that gene in humans? Okay? Which is the one that's most similar? Is it really a lot similar still? Does it do the same thing? Maybe it has something to do with regulating how fat humans get. Okay? And this is why we use sort of this idea of evolution and homology. Okay? Sequence search. Evolution sort of justifies why we do sequence search. Okay? If we assume that instead every species was created individually and the genome sequences are wildly dissimilar, just making it up in a, you shake up a test tube and there's your genome sequence for tigers and cats and people, there'd be no similarity, there'd be no reason to do sequence search. Because we have this evolutionary process, sequence similarity search is well motivated, okay? And that's why it's a very important problem that we're going to study in here. Yes?
Is it the case that very because of evolution, the size of the organism has become increasing?
Okay, the question is, does the size of the genome, the size of the genome increase? You mean the organism or the genome?
The
Organism. So the answer is it's not necessarily true that organisms get bigger in history. I mean, if you go to a museum, you see the dinosaurs. The dinosaurs were very big, and they're not with us anymore. Okay? So what probably happened is there was a period of time when being big was very good. Okay? And that you could step on smaller dinosaurs, and if you got bigger than your neighboring dinosaur, you had more babies or you survived better. Okay? But then somehow there came a time when situation changed and it was not as advantageous. Okay, like there's this vision of an asteroid coming and splatting Earth and making it dark. And now if you're a big dinosaur, you needed a lot more energy, let's say, than if you were a little dinosaur. Suddenly the conditions have changed. All this evolving you did to become big is now disadvantageous. Now the situation benefits the little animals. Okay? So there's certainly no monotonicity about size of the organism. Does that make sense? Yes.
So, when you say that both had this common ancestors,
Yes.
and these are two separate parts, but why not again why don't we find the halfway to humans?
Okay, so the question now is, what you're saying is, why isn't there a species today that is halfway between human? Okay? So the short answer, okay, so you're telling me basically, okay, so if I understand the question, I think it's a good question. You're telling me that basically here we have an ancestor to us both, okay? This ancestor was neither a mouse nor a human nor a chimp, okay? But at this point, there are two living species that are descendants of this, right? There is the human and the chimp. Now, probably what happened is there were many other intermediate species that split off. For example, about 500,000 years ago, there was a common ancestor. There was a Neanderthal man, split off from human, as another caveman, right? And so there was a common ancestor here. There were cavemen, Neanderthal men, and then some reason they went extinct. Why was it? Maybe the world got colder and you had to be smart to survive. So only modern humans survived, right? Or maybe you had a group of intermediate chimps, not something that looked more like a monkey to you, but it wasn't good at climbing trees. It was good in grasslands. And suddenly the place where they lived grew a lot of trees. Okay, the ones with a tail did very well. They had lots of babies. They lived long. And the other one went extinct. So the way to think about it is there's a small number of surviving species. There are a lot of intermediate life forms that were there and sort of went away. Yeah.
Is that part of the natural selection?
That's part of the natural selection, right? And that somehow what's good for a while isn't that good isn't good in all conditions. Okay? And you would expect that eventually for a given environment, a currently best, current champion, okay? And that's what lives, and it drifts and evolves and stuff like that. Any questions about that? Yes?
Is copying error the only difference why the genomes differ? Is the only reason why?
Okay, so again, the question is copy error the only reason why genomes differ? First of all, again, in biology, the first principle is nothing is ever always true. Okay, so when you say, is it the only way of doing it, the answer, whatever the rest of your question is wrong. Okay, the answer is no. There's always other things. But what kind of things matter? There are these what things kind of matter? Again, I think that there's these mixings of different populations, and by these mommy and daddy recombinations. There are these point mutations that are sometimes important. Okay, and in fact, if you look at what is the difference between humans and chimps, a lot of it is a question of a small number of mutations in how certain genes are turned on and turned off. Like there's one gene that seems to be linked somehow to speech, and this has gone rapid evolution between us and chimps. Oh, that's why you don't listen to chimps. They don't say very much. Okay? So the answer is, I would say a lot of them are basically, I would say it's some form usually of recombination or mutation are the main, let's say, changes of genome operations. Sometimes they are very catastrophic mutations that happen. So one type of mutation that's actually probably pretty common is that your children, let's say that there might be a recopying of a section of a genome. So you can imagine, like, here is your genome sequence. Maybe there is an evolutionary event where this sequence gets copied and stuck again at the back. Okay? Now you've got two copies of each one of these genes. Okay? That's a common kind of a mutation. Okay? And often this will kill the organism. But sometimes this is good. Now you've got two copies of the genes. Maybe making more of these proteins is a good thing. Okay? Maybe it doesn't matter, in which case it's not a bad thing, but it might be, might just happen to be passed on anyway. And then this copy can evolve into other things. So evolution works in funny ways, but the way to think about it basically is that there are some event happens, you are different than your parent. Okay, you are different than every other person in this room. If you are more successful, your genes will be passed on, and hopefully the whole population will come to look like you. Okay? It would be good if you're successful enough that you come back 100 generations from now and the whole planet looked like you. Okay? That's what, in some sense, evolution is wanting. You're wanting to, your genes want to do. Okay? Probably won't happen, but any questions? Okay? Any more questions about evolution? Again, it's a very important process. And this notion of homology search, which is really what we're going to be talking a lot in here about sequence comparison. The interesting thing is how far in evolutionary history you can detect similarity between organisms. So we were talking about yeast, okay, last class. That's an important model organism. A lot of important genes, you may say, what do I have in common with a yeast? Okay? The short answer is a lot of the, yeast has about 6,000 genes. You have about 25,000 genes. But a lot of the things that are essential for things like cell division, okay, which you do and yeast does, those genes are basically the same thing. And so you can detect sometimes sequence similarity over hundreds of millions of years difference, okay, if your homology detection algorithm is good enough. And that's what motivates us looking for, doing sequence searches. Any questions about that? Any questions about search, why it's a good thing? Let's see. Okay, hold on. Oh, good. Okay, good. Let's move on. Let's try this. Sorry. Oops. No, trouble, no. Go away. Okay, let's just see if I can do this. No. Okay. Timeout. Okay. Oh, good. Okay, good. Let me try to move this forward. Okay, sorry, we're almost. I'd like to think we get back there. Maybe we'll evolve a species that's better at doing this. My ability to get tenure would depend upon how well I could use this thing. Okay, you would evolve professors that are better at this. But okay, let's just keep going around. Okay, let's try this. Okay, so anyway, like I said, evolution is an important thing for us. Okay, we will be talking in here a lot about evolutionary trees. Okay, which again, I showed you a picture of a tree. phylogeny is sort of the word we will use for sort of an evolutionary tree. So you'll hear me talk about that at the end of the semester. But what's interesting is we can sort of use data about genes to try to reconstruct evolutionary history as well. This is actually kind of an interesting phenomenon. If we agree that all the current species evolved from a single ancestor, and we look at all the leaves of a tree, okay, here we've got 5 species, it looks like, in this leaf tree, okay, and we now take a look at the genes, okay, in different species, and we compare how similar they are. We would expect that this species, let's call these species A, B, C, D, and E. We would expect that the genes that are most similar to E's are probably D's, because they had a common ancestor most recently. The most similar ones to B are probably C's genes. If we look at A's genes are probably going to be more similar to B and C's genes than they are to D and E's genes. Does that kind of make sense? Because there was a time when they were the same, they've drifted apart. What this means is, just from taking a look at the genes and looking by how similar they are, you can hope to try to reconstruct the history of how they emerged, right? Here we've got, on humans, what do we know? We look around our world, we have people, we have squirrels, we have chimps, we have tigers, we have lizards, okay? What species evolved from what? What was that tree? The interesting thing is that you can go through and by looking at the similarity of genes, you get distances between species. From that, you can hope to try to infer something about the topology of the tree. And reconstructing this history of evolution is a very interesting thing. It's what we're going to talk about the last couple of weeks of the semester. Okay? Any questions about that? Does it make sense? Yeah, question.
I noticed that you keep on drawing binary trees, but it's not necessarily binary trees, right?
Well, the question is, are phylogenetic trees binary? And I guess what I would say is, in principle, they probably are, okay, on a certain level. But recognize, okay, so let's think what is happening. Each node, each leaf here is a species, okay? And what is happening is that there is an event in time that causes a population to sort of diverge into 2 directions usually, okay? So what I kind of am imagining is that, you have an instant in time when there was one species, okay? And then suddenly there was a mutation, one of these duplication events that I talked about. And now they're split off a whole other population, which is quite different from the first. If you kind of picture it as being a new creature being born that is wildly different than the predecessor, that would suggest a binary split. There are the old guys, and then there is the new guy, right? So on one level, these things usually, quote unquote, should be binary trees, okay, on that view of the world. Sometimes they are not really binary trees because you don't have enough data about the emergence of what happened, okay? So you could imagine a world where if we try to reconstruct our past, we might not be able to know enough detail to know exactly what order closely related species broke off. And so sometimes these trees may not be binary. But I think that if you think about it, the biological process, if you really add all the information, binary is a pretty good model.
Well, I mean, it could be possible that there could be 3 things happen that...
Okay, it could be. It could be everything. I said nothing is always true in biology. I think of these binary trees, I think comfortably of them as binary trees, although they're not always binary trees. And there are other weird evolutionary things that happen that sometimes don't even cause them to be trees at all, okay? But I don't want to go there right now, okay? I think of them as trees, I think of them as binary trees, and I can get pretty far doing that. Yes? So what sort of happens is, what is a split? I picture a split can be several different things. One thing that a split could be is that there is a genetic event, and suddenly this population splits off and doesn't interact with the other one because, it's so wildly different. Maybe like in the case of a single-celled organism, right? You could imagine that it splits off. Another reason it might cause a split that's probably more common is that something happens with the environment. You could imagine a world like, for example, I don't know if you've heard about the theory of continental drift. I'm sure probably of you have already seen it. You could imagine a world where, what is it? Here's Africa, here's South America, or something like this. At one point, there's a family that's sitting here comfortably interacting, right? And then suddenly breaks apart. Water comes in here, okay? This side of the family can't breed with this side of the family, right? There now is now an ocean between them. And now they're going to go their separate ways, and right now they're still genetically the same. But if you come back a million years later, you would expect the populations will evolve in different ways, okay? And after they separate for a while, you've got new species. And that's probably a more common source of speciation, these kinds of things, okay? Any questions? Okay? So anyway, evolutionary theory is an important thing. For us, we are going to believe in evolution. Some people, have issues with this. Okay? In this class, we believe very much in the theory of evolution. The argument that, by the way, that if you want to say, why do I believe in evolution or something like this, it is one thing that is quite convincing to me is that before any of this DNA evidence came out, you had a large number. I mean, Darwin was before people knew it. Didn't know how to spell DNA. DNA wasn't discovered until, 100 years after Darwin published his book, basically. People were still, taxonomists were people who studied organisms and had tried to say, humans and chimps, they look pretty similar. Why do they look similar? Well, by looking at all the features, you could see, geez, humans and chimps have a lot in common, okay? Tigers and humans don't seem to have as much in common. And taxonomists had worked out sort of these phylogenetic trees based on physical evidence, okay? Where were there bone openings in the skull and things like this, okay? And based on that, they had trees. The thing that is interesting to me is, to a large extent, how well the DNA seed trees that people built afterwards matched the trees that taxonomists were using. Okay? I mean, they didn't match perfectly. There's some substantial differences. But it is pretty clear that the DNA evidence supporting, phylogeny to a large extent supports the stuff you would get on just taxonomical grounds. And it's sort of an independent confirmation. That's sort of it. Okay? So from here, we're going to believe in evolution, and it's important because it's going to give us reasons to study evolutionary trees and sequence search. Any questions about that? Yes? There are largely, as I understand, first of all, recognize I'm wading into territories, that I don't know. Largely, I guess there are religious and cultural evidence, okay, or arguments that make some people reluctant to accept this line of reasoning. The scientific evidence seems to be pretty good in many ways that evolution works this way. And certainly as a model for what we're doing, this is what we're gonna do. Okay? Any questions about that? Okay? Fair enough. Let me now try to go, see if I can move forward. Boom. Click here to restore writing. No. Let's see if I can, is this it? Bingo, this may be the answer. Okay.
On top of the screen is next and previous. I don't know if that one.
You would think this might make a difference? Let's take a look at it. Boom. Boom, that does look good. Okay, good. Okay, let's try it. My keyboard also seems to have next and previous, but it doesn't seem to work. I'll try to remember to do that. Thank you.
Sometimes there's arrows too on the laptop.
Yeah, the arrows don't seem to be doing the trick, but this looks better. Okay, any questions about evolution or life or anything like that? Okay, so what I'd like to talk about now is not so much life. We've talked a little bit about how life is organized, and that's important to govern it. The last part about the, let's say, biological preliminaries that I'd like to talk about are biotechnologies. So the other thing that drives what we're doing is not just how life is organized, but that there are amazing technologies that people can use in the lab to manipulate DNA molecules. So why do we study DNA molecules? Well, it does encode all of, the information to make an organism. It clearly evolves. Lots of good things to study. But the main reason we study, for example, for DNA proteins, molecules, is that there are lots of technologies that enable you to manipulate DNA molecules, to alter them so you can experiment with them, to measure properties of them. And it's important to understand what some of these technologies are. Because a lot of the stuff we're going to talk about, when we talk about sequencing and how the genome was sequenced, that is a technology question. How do you sequence a genome? How do you read a DNA sequence? Okay? And in order to do that, you need to be able to do certain kind of technological things. There's certain elementary operations a molecular biologist can do in a lab. And it's important to know what they can do and how you can put them together to do other things. Okay? Any questions about that? Again, I find these technologies very amazing to me in the same way that I found VLSI fabrication an amazing thing. So, in the old days, I'm an old computer scientist. We used to take courses in digital logic, and we used to, take courses. I took a course in semiconductor fabrication. And you could either view a computer chip as being this thing, it's here, it's amazing. Or you can view it as this amazing thing that, my God, they wrote these things microscopic. They wrote down, 100 million transistors at a microscopic size on this thing, and they didn't get any of them wrong. Okay? And that, to me, is an amazing thing. They're able to manipulate matter at this scale, this accurately. And likewise, what they can do with DNA molecules is amazing, and we should respect that. And that is what a molecular biologist is and why these are interesting people to talk to, because they can do amazing things with molecules. Yes?
You said short DNA. How would that compare to the DNA of us?
Well, okay, I'll go through these things. Let's now talk about, what I want to talk about are a couple, actually 3 different technologies that I want you to sort of believe or understand them well enough to believe that they work so that we can make things out of them. That's kind of, I guess, the level that I want to make sure about. So the first of these technologies, I'll say, is DNA synthesis. Okay? So basically, one thing you can do that molecular biologists know how to do now is to build machines that will synthesize DNA molecules to specification if they're short enough. So let's say that you want a DNA molecule, ACGTT, okay? Let's say where short is about, let's say, 50 bases, something like that, okay? You can actually go and make these molecules. Actually, people don't make them. What they can do is call up a company, give them a credit card, and say, here is $50. Give me a test tube with this molecule synthesized, many copies of it. And they'll get it by FedEx the next day. Okay? So the interesting thing is that short molecules you can synthesize 20 bases, 50 bases, something like that, quite cheaply and quite routinely. Now, how does that work? The way I think about it is they make molecules. Remember, we agree the molecules are made of A, C, G, and T's, right? What they do essentially are build molecular building blocks of the form a sticky thing, a letter, and a cap. Okay? So you guys remember in chemistry you have chemicals, you can bond chemicals to other chemicals. That's the kind of thing chemists do, right? So now imagine that you could have you could buy a bottle of A's with sticky things on one side and caps on the other. Okay? Now, and the same thing, we would also buy C's, G's, and T's with that property, right? How might I grow this molecule? What if I have something that sticky things want to stick to? The other side of Scotch tape or something like that. And I pour a bottle of this stuff to it. What's going to happen? Well, the sticky A is going to stick to the sticky thing. And there's going to be a cap that doesn't let anything stick to it, right? So I'm only going to get one A sticking there. Does that sound reasonable? Now suppose I do a chemical reaction that knocks the cap off. Okay, and what's left is a sticky thing. And now I pour some of my C goop over it, right? What's going to happen now? Okay, this molecule, the C sticky thing, is going to bind to the sticky thing, and I'm going to get a cap on it. Does that make sense? And by doing a sequence, if I want to build a molecule of length 50, If I do a sequence of 50 bottle pores, followed by unblocking operations, I will build up a large number of these molecules, these chains, to exactly the specification I want. Yes?
Why not you keep on doing this and build a bigger one?
Okay, so why can't I build a bigger one? Well, the big reason is because, again, these are molecules, and what they do with molecules is an amazing thing, and there's errors and things like that. You could imagine, for example, if I pour this thing over, I have a lot of these guys. Maybe for some reason, none of the sticky stuff is going to stick in one round. Does that make sense? And then maybe I might get one where I didn't have a C, and the next thing was a G. So there are errors potentially in this process. And if I do this process too many times, the odds that I get anything that's perfectly right is very small. Does that make sense? You know, if let's say 1% of the time I am going to make an error, if I do this, 50 times, most of the molecules are probably still right. If I do this 100 times, most of the molecules are wrong. Does that make sense? So that's sort of an argument why short things work. Okay? Any questions about that? Okay? Yeah.
Is this, I mean, like A, do they fit in some kind of solution? I mean, how do they?
Well, this is their business. Okay? I mean, on some level, there's a lot of chemistry, there's a lot of stuff these guys know. Okay? But on a conceptual block, I can build, the thing you should take away is I can build short molecules to order. Okay? And get them by FedEx. Okay? Any questions about that? And it should be plausible that, yeah, maybe I can build them in this way. But I've obviously got to know some chemistry in order to do this. Okay? Any questions? Okay? So that's one technology. A second technology that is more important but depends on the first is a technology called PCR. Okay? Or the polymerase change reaction. Let's just call it PCR. And what PCR does, it's a way to make copies of molecules, okay, or portions of molecules based on what it sits between. So let me just go through this quickly. What PCR essentially does is it lets you do the following. We said that DNA sticks to other DNA, right? There's this hybridization thing, this reverse complement, A's stick to T's. Suppose I have a DNA molecule here, and I want to make copies of just this portion of the DNA molecule here. Just say it's a gene. I want to make lots of copies of that part of the molecule. What can I do? I can synthesize a sequence that will bind here, and essentially a sequence that will bind here. There's some double-stranded issues here. I don't want to go through too much detail here. I can bind there. Okay? And then basically what I can do is tell it to copy between where this sticks and where that sticks. Okay? And basically there is this thing, it starts here, you say start growing, it copies from here, and let's say it falls off here. Okay? And it does the same thing on the other strand. Copies from here, copies from here, because you put the complementary, they call these things primers, these starting sequences. And then if you keep doing this thing over and over again, you can basically get the number of copies to grow exponentially in the amount of time you run this thing. So if you start with one copy of a molecule, if you know what pattern is before it, what pattern is at the end of it, you synthesize these starting primers, you stick it in a PCR machine, and it's going to make many, 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 many copies of the molecule between there. Okay? That is what PCR is. Okay? It's essentially a way to copy things by position. Okay? Not position in the long sequence. Copy it by context, I guess, is the right way to think of it. Right? You don't say copy base 963 through 752. You say copy the stuff between this sequence and that sequence. Okay? And that's what PCR is. Any questions? Yeah?
Does it also have a limit on the length of the...
Yeah, so this copying thing only will work for things typically, let's say, about 3,000 bases long. Sometimes they can push it a little more, but it's in the order of a small number of thousands bases, not millions or billions of bases. Okay? But it's an amazingly useful thing to be able to do. So if you want to take a gene and copy it a zillion times, you can do that. So let's say you wanted to study a gene, okay? Maybe to sequence it or something like that. How would I sequence your hemoglobin gene? Well, I take some sample of your tissue. I've got 3 billion bases of DNA to deal with. How am I going to fish out your hemoglobin gene? Well, what I could do is say, gee, you're a lot like this other guy, because you're a person, you're within one, you're almost identical. I look at the sequence in him that is just upstream of the gene I care about. I look at the sequence in him that's just downstream from what I care about. And I say PCR it. And now I make a zillion copies of just the sequence that I care about. Now I have a lot of DNA to study. Okay? And that is why PCR is an important thing. Any questions about that? Any questions about what PCR is? You can think of it as copying. The exact mechanics of it are not that complicated, but I don't want to go through it. Okay? But PCR lets you copy by context. Any questions? Okay. So biologists, molecular biologists, as far as I'm going to do 3 things. They can synthesize things by cutting a check to the company. They can do PCR of copying. And they can do one other thing. Okay? Let's see if I can, oh, right. You say I can click on this. Very good. Okay? And they can do one other elementary operation. Okay? Okay? They can do something to measure the length of molecules. Okay? And this is something that we call electrophoresis. How might you measure the length of a DNA molecule? Okay? If you think about it, this seems like a complicated thing. Why is it? You can't take a molecule and put it on a scale. Why can't you put a molecule on the scale? Well, for one thing, you can't pick it up. How are you going to pick it up and find it? And the other is, how much is the scale going to go down? So biologists have figured out an interesting way to measure the length of molecules. Okay? Basically by using a tub of Jell-O. Okay? How many people know what Jell-O is? Gelatin. Okay? It's this goopy stuff, right? So suppose, let's say, I wanted to find out how much you weighed. Okay? And all I had was a big tub of Jell-O, a rope, and 4 strong people. How could I figure out how much you weigh? Okay? One possibility is I tie a rope to you. Okay? I have the 4 strong people pull the rope. But first I threw you in the tub of Jell-O. Okay? And I say, pull on these people for a minute. Okay? What's going to happen? As these people start pulling and tugging on you, okay, slowly you're going to get dragged across the Jell-O. Does everybody agree with that? How fast will you get moved across that Jell-O? What is it a function of? Okay, depends upon the weight of the person being moved through the Jell-O. Does everybody agree with that? Suppose I have this really heavy guy, okay, who weighs twice as much as you. And I say I have 4 equally strong people. Pull this guy through the Jell-O, right? What would we expect to happen after a minute? Okay? You would have probably gotten this far, right? The heavy guy might only have gotten half as far. Does that make sense?
It also depends on orientation.
Okay. So okay, let's just think what we mean by that. You say it depends upon surface. What does that mean?
Yeah, like how wide, let's just say the person, maybe not the weight, but just the
Okay, I see. So you're telling me that it's not just a question that is I cut through this Jell-O. Okay, it's not just the fact that I'm heavy. I'm not a point mass, but I'm flailing here. Okay, how much I'm flailing will resist me how much I go through this Jell-O, right? If I am wide but light, okay, I will probably not move through the Jell-O as quickly. Is that right?
Yes.
That seems perfectly plausible. So the answer is this a good way to exactly measure how heavy you are? No. But is it a way to get an approximate sense that this person is probably about half as much mass as the other guy? The answer is yes, right? So the answer is this sort of gel idea gives you a rough way to measure how heavy something is. Okay? Not perfect. Okay? But molecules are very small. And to measure the weight of something, you can do a pretty good job under certain conditions getting pretty accurate measurements. Okay? If you control the jello carefully, if you do everything the way that it's done properly. Okay? But we can't expect perfect answers. That's one thing that's certainly true. We should understand that we can't expect perfect answers to anything these guys do, because it's complicated doing things with molecules, for goodness sakes. Yes?
What if you replicate it so many times that you get a mass that you can easily weigh, and then you divide it by the number of times you replicate?
So you're saying, what if so one way that you might be saying is, what about if I take advantage of the fact that I can make lots of copies of it? Maybe that is an interesting reason to make copies of something. So maybe one way, maybe I get a more accurate answer because I have a lot of copies of something. Does that kind of make sense? That if I, instead made several copies of him, and we dragged each one of them separately through a fresh thing of jello, if we average how far it got, that probably gives us a better answer, doesn't it? Right? So suppose I want to figure out how long a gene is, let's say. It makes sense that the first thing I would do is I would copy it many, 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 many times, right, by using PCR, okay? I will stick it in my tub of jello. It actually is a gel, okay? And instead of using the strong dyes, I'm going to take advantage of the fact that these molecules have a charge on it. And we know that positive molecules attract negative molecules. Does everybody remember that, sort of? So you could imagine that these molecules, if they're positively charged, and I have a negative charge on the other side of the end of the pool, this molecule is going to want to wiggle towards there. Is that right? And they're going to fight their way through the jello. Okay? And if we wait a certain amount of time, some of the copies will get luckier and move a little further on than other copies. But we would probably expect like some kind of a distribution of molecules there, a smudge on the gel, right? And maybe the center of the smudge is the best estimate for how big the molecule is. Does that make sense? Okay? And so the third technology that these guys can do is that they can measure they can measure the length of molecules to some level of resolution. Okay? Any questions about that? Okay? So these guys can copy molecules, they can measure the length of them, and they can synthesize them if they need them to a certain length. And that's a large part of the tricks that any molecular biologist has. Okay? They can't do that many things. Okay? But that's what they can do. Any questions about it? Okay? Yes?
Yeah, about the graph. So that the graph is right, and the top, the middle of the graph is the exact weight that we are looking for.
Yeah.
But that would not be the average thing, right? I think the average weight that we are getting would be more accurate.
Okay, so the question, I think, is you agree that if I have a lot of copies, and maybe I'll make all these molecules radioactive or something so I can detect them. I have to see these molecules, right? How do you see molecules? You look through a microscope? That won't work, right? But you could imagine that if I put something on them to make them radioactive or make them give off light, fluoresce, okay, I will be able to see a smudge corresponding to this group of molecules when it walked up. And now then there is a certain amount of analysis and image processing that needs to be done, if you wish, to convert the smudge to a length, right? How might I convert the smudge to a length? Well, part of it would depend upon having a reference standard, right? It would be good if I wanted to figure out how heavy he was, if I knew that this other guy weighed 300 pounds before I dragged him through, right? Then I would know that if the 300-pound guy got up to here and you got twice as far, well, then I know you weigh 150 pounds, right? There's got to be a conversion between smudge distance and weight, okay? So there's a certain art to this, a certain science to it. But in principle, this gives you a way to measure the length of molecules. Any questions about that? Okay, so these are the three basic things that yes?
So we can do the DNA sequence, but we cannot measure the length, correctly?
Well, No, no, no. So what we can do is, the question is, how can we sequence things when we can't measure length correctly? As I will talk about when we talk about DNA sequencing, it turns out that the way you sequence DNA actually is by doing PCR of what you want to, making copies of what you want in a clever way, and then measuring length. Okay? So the answer, it turns out, is that if you're measuring short lengths, you can do that pretty accurately. The longer the lengths are, the less resolution you get. Okay? So it turns out that DNA sequencing machines, if you said, how does an old-fashioned traditional DNA sequencing machine work? It does nothing more than make copies of the molecule that you want in a clever way that we'll talk about, and measures the length of each of these molecules. And there's going to be a way you can determine the sequence by the pattern of lengths. Okay? So the short answer is that you can do this accurately enough to do sequencing for a certain level. And we'll talk about that later on. Okay? So we'll talk more about how sequencing machines work in this way later. Okay? But it gets to the point that I want to say, by just being able to, you may say, these guys can't do very much. All they can do is copy molecules and measure length. Well, if you copy molecules, it's like saying, well, what can these computer scientists do? They can only put program instructions together. But if you put the instructions in the right way and you do it, you can do interesting things. Okay? Any questions? Any questions about these three basic technologies? Okay? Just to clarify it, actually, I don't think I want to say clear. How do I say clear? Okay, good. I'm going to show you one other killer app, let's say. One other thing. You can do sequencing using these technologies. Let me just tell you one other thing you can do, okay, by just knowing how to measure length of molecules and make copies of it. Is there any news story in the news recently about where they use DNA identification? You've probably seen TV shows or something like this, where they identify the bad guy from DNA. Is there any story like this in the news now? You know, again, I saw Bobby Fischer, the chess master, right? They wanted to figure out if he had a child or something like that. How were they going to tell that he was the father? Okay? Somehow, what they'd like to be able to do is to essentially, again, with paternity testing, it's a little bit more complicated. But you can imagine the basic problem of this. I give you a drop of blood, okay? And I want to now figure out if that blood belongs to you, right? There's a drop of blood on the floor where somebody which the murderer left after they had cut you with an axe, right? And somebody thinks that you did it, okay? Okay, maybe you were killing me in response to being upset at being dragged through the Jell-O or something like that. Okay? How can they tell that that blood belongs to you? Okay? It turns out that basically these things work using just copying and length molecule measurements. Why is that? Turns out that on your genome, there are certain places where it's in some sense in the junk part of the DNA where it doesn't matter very much. There happens to be very boring parts of the sequence that repeat over and over again. AT, 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 AT. Okay? And I happen to have 57 ATs in this spot. And you have 47 ATs in this spot. Okay? Everybody has at a certain point on their genome, has this highly repetitive region. And there tends to be differences. It doesn't have a function, really. Okay, but it's a historical artifact. Okay? What can I do? Okay, if I want to try to convince myself that this blood is you. Okay? I could take a sample of blood from you, and I could now say, okay, I'll take the upstream region, and I'll synthesize that primer, downstream region, synthesize that primer, make a zillion copies of the molecule between there, and measure the length of these things. And suppose I discover that yours is 57 repeats long, right? I now take this one drop of blood, which is precious, right? There's only one drop. I can't get the murderer to leave more, right? Okay? But I can copy things, right? I now take this blood drop and I copy between those same two starting and ending points. And I measure the length of that molecule. Okay? And it comes out to be 57 long. Okay? Does that mean he's the murderer? Not probably. Not yet. Okay? Maybe. Because a lot of people in here probably, a lot of people probably have 57, right? But what if I go to another place on the genome and I copy it here and I give a separate start and end point? And here he comes out with 63 copies. And the blood comes out 63 copies. Now it's looking worse for him, right? If I do this 10 times, okay, and each time it looks like it's the same length, I think we got the guy, right? Okay? Yes? So twins are a problem, okay? In principle, identical twins should have the same genetic, so that might be your defense. You may want to be his defense lawyer, okay, and say that there's a long-lost evil twin here, okay? But so there are still the questions of how unlikely is it? The real question you're asking with me, what other possible explanations? If he doesn't confess in the light of this evidence, you have to start asking how rare is it and stuff like that. And that starts to be a population statistical study and all these kind of things. But does everybody see how just being able to measure the length of molecules, okay, and being able to make copies of things, okay, I can do amazing things, okay, yes? Let's say that this is roughly, let's say these variation parts, let's say I'll put that into 1/1000th variation of difference, okay? So there are these small places where there are these repeats, okay, that sort of these stuttering copying issues, okay? And those seem to be good for doing this kind of identification. Any questions? Yes?
Are these good, are these copies, let's say 57 is the number you try 10 times or 12 times, is it enough to satisfy the rule that yes, these are pure?
So you're saying how convincing is it that I have 10 different markers and all 10 of them are the same? Well, you can imagine that they've probably done studies in the population. How many people have, what is the statistical frequency of each marker in the population? A good marker is one where there is a lot of variation in the population, right? And if you can argue that these are independent, and if you argue that there's a 1/2 chance, let's say that there's a 1% chance that you and I share 57. 57 only happens 1 out of every 100 times. If I do this 10 times, it's 1 out of 100 times 1 out of 100 times 1 out of 100, the odds become very, very low. Okay? And so the argument is that this now then starts to become very convincing identification. You know, there are problems. Maybe it's identical twins. Maybe it's something different. But you've got to argue that in court. And this is why, you know, your lawyer does not want you to come out the winner on one of these tests. Yes?
And these markers are consistent just on the left.
Right, so there tend to be places on the genome where there is a lot of this repetition and variation in between. Okay, and so they've discovered these places, and they happen to be places where there are a lot of these repeated patterns and a lot of length difference. Okay?
So at least the markers are pretty much the same.
Right, so the bounding regions are identical enough that the PCR primers can hybridize to it. The repeated stuff is in between there. Okay? And that's what you would need in order to get this kind of thing to work. You want the bounding stuff has to be good enough to amplify. Okay? The stuff in between has to be variable enough to make it interesting that you know, the number that you get from it. Any questions about that? Or any questions about this whole DNA technology business? Okay? Good. So what I'd like to, okay, I see, right, next. Okay, so any other questions about biology? At this point, I'm going to say we all know enough biology to take this class. Again, I encourage you to read the chapter and, you know, review this stuff and get a better sense about it. Because, you know, it's important to understand it and see what happens. But conceptually, this is the biology we need to start the course. Any questions? Okay? So what I'd like to do for the rest of class and starting next class is to introduce computer science for biologists. Now, the biologists in this room, meaning the non-CS people, you should have seen the ridiculous questions the CS people have been asking. And so I don't want anyone to be shy here about asking a question. Oh, what is a computer? What happens if it breaks? Any of these things. Any question that you ask is fair game now. Recognize that the questions that we work this way. So I do want to encourage equal. But we're going to talk about some interesting issues in computer science, and I expect there will be questions from the other side too. So don't everybody get it wrong with it. Okay, so in computer science, what the main thing in computer science that computer scientists, or at least that I will say I study, and the computer scientists like me study, is we are not so much interested in properties of computers. We don't care about hardware. We don't care about software. In principle, what we care about are algorithms. Okay? And what is an algorithm? An algorithm is a procedure that always provides the best possible solution to a well-defined problem. So and I'll give you an example of a well-defined problem on the next slide. But basically what our business as a computer scientist is we wander around looking for problems that are well-defined enough that we can define algorithms that correctly solve that problem and do it as quickly as possible. That's sort of what our business is. In this class, what our main business is, we're going to be looking for problems that are well-defined and that we are going to try to solve them always so they're correct, okay? We always get the optimal answer back when we can, and in a way that is as efficient as possible, okay? That is what computer scientists do. Any questions about what computer scientists do? Now, what I want to distinguish between are as sort of a fundamental thing that is, surprising unless you've lived in this world. The distinction between a heuristic and an algorithm. Okay? An algorithm, as far as I'm concerned, has a well-defined problem it's solving and always guarantees the right answer. The heuristic is something that tries to solve a presumably well-defined problem, but gives me usually a good enough answer. Okay? But no promise it's absolutely the best. Okay? So, a lot of times in here we're going to have a problem. And there's going to be a lot of common sense things you might think to do to solve it. Okay? And maybe these will behave well in practice or usually do well. Heuristic is one that may give me a good answer but has no promises. An algorithm is the one that gives me promises. And so what we're going to be doing in this class is for each problem we come in, asking, is there something that will always guarantee me a good solution? Or am I just trying my luck and trying to do something that makes sense? That's a heuristic. Any questions about the difference between a heuristic or an algorithm? But this is a fundamental distinction, and we'll see this again a little bit later. I suspect probably next class. But we'll see this issue, because that's an important thing. And so what I, as a computer scientist, am doing is I go talk to my biologist. My biologist says I have a messy problem. I just say, oh, what do you want to do? Show me the genes in this sequence, okay? And I will say, well, that's not well-defined yet. Give me a well-defined. What is a gene? Well, a gene codes for a protein, okay? What is a protein? What does it mean to be a gene? What separates a gene from a non-gene? And I will keep asking you until you give me a deterministic thing that tells me exactly what I'm looking for. If you tell me exactly what it's looking for, your problem is now well-defined, and now I can seek an algorithm for it. If you tell me, yeah, just give me something that's interesting to me, okay? That's not a well-defined problem. I obviously can't guarantee a solution to it, okay? But in general, we're going to be looking here for well-defined problems which are related to the real messy problem, okay? And for that, we will seek an algorithm. Sometimes we won't be able to do that. We'll admit our sin and live and settle for a heuristic. Any questions about that? Any questions? Heuristics versus algorithms. Okay. Let's look at an algorithm quickly, okay? Or a problem or an example. Here is an example of a problem that is a well-defined, important problem that we will be looking at actually in this class in some detail. Is the problem of exact string matching. A problem is specified by an input and an output. Okay? The input says I am given as input a text string T. Okay, here's T, and it's A, C, G, T, 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 C, whatever, dot, dot, dot. And a pattern string. Let's say G, A, T, T, C. Okay? That happens to be a little pattern that's of some biological interest. It's a restriction site. Okay, restriction ends it, cut it. And I might ask myself, as an output question, given this string, and given the text string, and given the pattern string, where is the point in the text string where this pattern occurs? That is the output problem, okay? And I claim this is now a well-defined problem. I've specified exactly what my inputs are, a text string of length n and a pattern string of length m, and I want to know where in the text string does the pattern exist. Does the pattern exist if there is such a place? Okay? Any questions about this problem? Okay? How many people see this as a well-defined problem? Okay? And this problem has an answer. If I give you any string and any text and any pattern, this problem has an answer. Either Skiena is in the Bible or Skiena is not in the Bible, right? Does everybody see that? Okay? It's the same principle here. The answer is no in that one. Okay? Any questions? Okay? Good. Okay, so that is what I mean by a problem. A problem is a well-specified thing. What do I mean by an algorithm? Okay? Here is, I believe, an algorithm for solving this problem. Actually, does anybody have an algorithm in mind for solving this problem? Here I have a text string. Here I have a pattern. Okay, again, I'm writing it as a star, but here is its letters. Okay, G A. Sorry, it's not right. Okay. Anyway, it's a text string. Okay. How can I find whether a given text string T here has a particular pattern P? Can anyone suggest an algorithm for it? Yes.
Simplest way would be to just start at a particular offset and check for len loop 3 and see if they match.
Okay, so what you're saying is, I see. So you're saying that here's my text string, here's my pattern. Okay, P. For some reason it doesn't like me anymore. This pen, I'm gonna break the pen. Okay, my my pattern. I will stick my pen. I don't like this thing. Okay, so what you're saying, and maybe I'll do this on the board. Okay, here I've got a text string. You know, G A T C, G A T T C. Okay, this is my text string T. This is my pattern string is going to be G A T T C. Okay, how can I tell whether this is here? You're saying maybe it starts in the first spot, right? And so I can go and say G, yeah, A, yeah, T, yeah, T, yeah, T, C, mistake, right? Does everybody see that? So it doesn't exist in the first spot. Start now at the second part of my string and say, is there a G? No, it doesn't work, right? Third character, is it start with a G? It doesn't work. That can't match, right? But when I get over to here, G, A, T, T, C, ding, it's done. Does everybody agree with that? So that is really a procedure. That's what I say I am doing over here. Okay? If you look at the procedure on here, okay, I am saying basically I is the starting point in my string. For every possible starting point, align the pattern, okay, and I'll have a counter J that starts at 1, and ask, is the first character of the pattern equal to the I plus ith position of the text? If it is, keep going. Now try the second position of the pattern against the next position of the text. Third position of the pattern. And I keep going around and around until either this test fails, meaning the pattern can't be there, or the loop ends because I've hit the end of my pattern. And the claim is under these conditions, okay, if I have tested the m characters of my pattern against the text, if they're all matches, then I have found what I'm looking for. Any questions about this algorithm, particularly from my non-CS people? Okay? Any questions? Yes?
Why did you start the sequence from 1 instead of 0?
Because I said that my sequence starts in the first position. Okay, so I have for some reason I have decided that the positions of my string are from 1 to n. If they were from 0 to n minus 1, I should start from 0. Okay? Any other questions about the algorithm? Okay? So this is in principle what we are seeking. We had a well-defined problem. We have given an algorithm now that if there is the pattern does exist in the text, it is always there. We find it. Clearly this is always going to be correct. How efficient is it? Okay, is going to be the next issue. And I'm just going to mention this for a second and probably talk more about this then. When we have an algorithm like this, we're also going to worry about its efficiency, okay, how fast it is. Actually, maybe I'll talk about this again. Maybe I'll talk about this next class. So let's say maybe I'm going to leave you guys here. So bottom line,